Hello, everyone, and thank you all for joining us for our weekly Nanaimo Bay education program, Nanaimo Talks. Today, I have Dr. Stacie Robinson from the NOAA Monk Seal Research Program, and she'll be presenting NOAA research ranges land to sea to understand risk of toxoplasmosis in Hawaiian monk seals. Be aware this is a pre-recorded presentation. She recorded it previously. So if you guys have any questions during the talk, please feel free to save it to the end or put it in the chat room, and we will get to them during the 15-minute question and answer. So without further ado, I'm going to share the presentation, and you guys sit back and enjoy.
Aloha! Thank you for joining me this evening and making time in your Thursday to learn a little bit about Hawaiian monk seals. My name is Stacie Robinson. I'm a research ecologist with NOAA's Hawaiian Monk Seal Research Program. And tonight I am going to be talking with you guys about a serious threat that faces seals here in the main Hawaiian Islands. So join me in exploring the environmental factors influencing toxoplasma infections in Hawaiian monk seals. First, let's talk about Hawaiian monk seals. The monk seals range throughout the entire Hawaiian archipelago, although only about 300 of the seals share the main Hawaiian Islands with us. The other approximately 1,100 inhabit the northwestern Hawaiian Islands, protected by the Papahanaumakuakea Marine National Monument. And there are some pretty dramatic habitat differences between these different regions. The northwestern Hawaiian Islands are primarily made up of small coral atolls and small sandy islands, though there's plenty of reef habitat for seal foraging under the water. And then here in the main Hawaiian Islands, we have much larger land masses, large mountainous islands with smaller beaches, but also some good foraging habitat under the water. And while the northwestern Hawaiian Islands do carry a legacy of human impacts in the area, some of the biggest threats to seals up there include ecological factors such as food limitation and shark predation. But here in the main Hawaiian Islands, seals have to share their habitat with a large human population, and so that exposes them to a very different suite of threats associated with humans, as well as the pets and invasive species that we bring to the islands. And so one particularly concerning invasive species is the domestic cats. Cats, or really their feces, are the key source of a dangerous pathogen, Toxoplasma gondii, and this is the pathogen that causes the disease Toxoplasmosis. So now cats have evolved with this parasite so that the parasite needs the cat because its sexual reproductive stage can only take place in the gut of a cat. And the cats are generally not badly impacted by the disease, but the disease can be very harmful to other species. In Hawaii, native birds and dolphins have died from toxoplasmosis, and toxoplasmosis can even be dangerous to humans as well. And so once a cat is infected, the parasite produces millions of oocysts. So an oocyst is basically a very small microscopic parasite egg. But a little different than the egg, it's extremely hardy, so it can persist for a very long time in the environment, and that means it can also be transported long distances throughout the environment. So why so much concern about this weird cat parasite for monk seals? Well, unfortunately, toxoplasmosis is actually a major threat to monk seals in the main Hawaiian Islands as the primary disease threat in the main islands. And it's actually behind anthropogenic caused trauma and interactions with fishing gear. Toxoplasmosis is actually the third leading cause of death for monk seals throughout the main Hawaiian Islands. Toxoplasmosis can have especially heavy impacts for the monk seal population. As you can see in this graph, toxoplasmosis hits adult females especially hard. This circle shows this large portion of the red bar. You can see the red bar is highest for adult females, so that's the age and sex group that is most heavily impacted by toxoplasmosis. You can also see that that red bar is the largest of all of the bars, or the biggest source of death for adult females. You can also see that because it can be passed from mom to pup, it impacts the number of young seals as well. And you see that it does impact all age groups. But one particular concern with those adult females being the hardest hit is that because those females represent the reproductive potential of the population, losing them not only takes out that one individual from the population, but it takes out that female's lifetime breeding potential. So this can really impact the growth rate of the population and the potential recovery of the entire population. So how do we minimize this threat of toxoplasmosis to Hawaiian monk seals? Well, we have 3 key tools that we can use. Those are emergency response, research, and management. And so first, let's talk about emergency response. And I want to share with you kind of a case study of one seal you might have heard about in the news earlier this year, RO28, also known as pohaku. She's an Oahu seal that came up sick with toxoplasmosis earlier in the year, and we put forth a pretty tremendous effort to try and save her. So I want to share a little bit about that story with you guys. Pohaku was first observed logging in January of this year, and logging is just our term for when a seal is sort of floating listlessly in the water, not really moving under her own power, not exhibiting normal seal movements and behaviors. And this is usually a sign that something is wrong. The seal is, doing this logging behavior because something hurts, something's not right, they're not feeling well. So this is a really important clue to us that we need to investigate something about this seal. And after observing this behavior for a couple of days, on January 22nd, we were able to capture her on the beach and bring her into the NOAA facility for assessment. And after giving supportive care for a few days, we got her toxoplasmosis test back and found out that she did indeed have toxoplasmosis. And so we knew we were in for a really big push to try to save this seal, which ended up being 10 weeks at a combination of some weeks at our NOAA facility here in Honolulu, and then a transfer to the Keikaiola Monk Seal Hospital over in Kona. And during this time, we really threw everything we possibly could at her care, doing everything we could to save her. So that included a number of lab tests, ultrasound tests, x-rays, trying to see what damage or what other issues she might be facing internally. It involved a number of medications daily. Here laid out on this table, you can see a bottle, you see pills, and that's Hope in that picture, and she's actually crumbling up those pills to put into that little measuring cup is holding fish mash. So the pills get crumbled up and put into the fish mash. There's also a paste down there at the bottom that is for an anti-parasitic paste that goes into the fish mash. And then all of that has to be force fed to the seal to get the seal to ingest all of these multiple medicines per day. So no easy task, as you might imagine. And in fact, as you might imagine, handling an adult monk seal for these treatments is very strenuous, both on the staff and on the seal. So we actually, while she was at the NOAA facility, we had additional vet techs and animal handlers from the Keikaiola Hospital coming over. Once we sent her over to their hospital, our vet tech helped them get started up for a while. So here you can see we're restraining the seal. I think this is probably just after we had given her her medications. We put the net over the seal's head for our safety as well as hers, and then the towel over her eyes kind of helps keep them calm. And then you can see in the back, you see a little bubble near the back, and that is some IV fluids going in. So a lot of work, not easy for the staff, and truly not easy for the seal, right? It's not easy for a wild animal to be in this very strange captive care scenario and not feeling terribly great. But we did see some progress with Pohaku. She did eventually start moving better. She started swimming around our pool. we did see some signs of progress, but a lot of ups and downs. And eventually, after those 10 weeks of really hard care, she did succumb to toxoplasmosis. So unfortunately, our biggest learning experience from this was that the emergency response is simply not enough. Like, we will always do our best to rescue any animal that is sick, and we will certainly keep learning and becoming better able to help animals learning even when we're not successful in a case like Oahu's. But we're simply not going to rescue our way out of this toxoplasmosis disease crisis. So our next step is to use research to try to learn as much as we can about factors influencing a seal's risk of getting toxoplasmosis. So in research, we ask questions, and probably the first question is, how on earth does a terrestrial parasite infect a marine mammal? And spoiler alert, the answer is not cats taking to the water. It's actually a pretty complicated question. You have to consider the pathogen that gets into the terrestrial landscape, the environment and environmental factors that might help transport that pathogen, and then the host of interest, the monk seal, that then is going to become exposed to that pathogen. And so thinking about these three factors, the host, the environment, and the pathogen, are what we call the epidemiological triangle. And so in tackling just one leg of this triangle first, our first effort is going to be to establish some basic links in this system. So the first foundational question we're going to ask ourselves is, are toxoplasmosis strandings more likely after a heavy rainfall or runoff event? Because if we see a relationship between these big water flow events and toxoplasmosis strandings, that might give us some clues that, the water flow, the runoff, is part of what's maybe washing some of these pathogens from the terrestrial environment and carrying them into the marine environment where seals can become exposed. And so we're going to try to answer this question using a case control study in which we're going to compare stream flow patterns preceding strandings of either toxoplasmosis cases or controls, which are strandings of others. First, let me tell you a little bit about the data that we use to do this. In order to characterize these major runoff events washing things from land to sea, we used the state of Hawaii's stream gauge data and looked at daily average stream levels. So this is the metric that we're going to use to measure the stream flow part of our equation, and I'll show you what that data looks like. The first thing we did was divide up the stream gauges across Oahu into different zones. So you can see different zones in different colors on this map, and that's basically so that each of our case and control strandings can be represented by a few different stream gauges to get a little bit more solid measurement of what's going on. And so then we plotted the data for all of those daily stream level readings for all these different regions. So you can see there in sort of the yellowish orange, all those ups and downs are showing the variation in stream levels on the west side. The different shades of blue are different areas of the windward side. And you've got the lines in gray represent the streams in town, and then green is for the North Shore. And so then in order to think about which of these levels are actually meaningful, what really constitutes a major water flow event, so we did a little analysis. You can see the bottom line there, that's the long-term average. So that's if you average all of the stream flow across all of the different days, that's the average stream flow. That next line where it says 1 SD, that is one standard deviation above the average, which basically means that's a pretty big darn water flow. And then you see the line that says 2 SD, or 2 standard deviations above the average. 2 standard deviations basically means this is a much bigger water flow than is really expected by the range of variation within the average. So what we did was we said any flow events that are above 2 standard deviations beyond the average, that constitutes a major water flow event, and we're going to see if these major water flow events correspond with either toxoplasmosis stranding cases or controls, or if it's all just random. So now let's talk about the monk seals we used in our study. We focused on Oahu from 2014 to 2020 because this is the time where we have the most toxoplasmosis cases, and Oahu is the island where we have the most toxoplasmosis cases. So we looked at 7 cases that occurred during this time, and this was all of the toxoplasmosis cases on Oahu other than pups that died in association with an infected mother. And then we looked at 10 control seals over the same period. And so a control basically means a seal that also was a stranding, a seal that died, but from some other cause. So these 10 were just seals that had a known cause of death. So we knew it wasn't toxo, but we knew it was something else, in trauma or drowning or other illnesses that we knew were not toxoplasmosis. And so we looked at the date and the location where the seal was first recorded as stranding in order to get more information about those cases and controls. So let's walk through some of this data together. So we're going to start with our first toxoplasmosis case. This was RO17, an animal that stranded on the northern windward side. And that black star represents the seal's stranding date. And then that red box is shading 6 weeks prior to that stranding date. And so within that red box, or within those 6 weeks prior to stranding, we're going to be looking at the stream gauge data to determine whether there were any major stream flow events in the short period before the seal stranded. And so since this seal was stranded on the northern windward side, we're going to be focusing on the blue lines. And I know with all the colors it might be a little bit hard to see, but inside that red box, you can actually see there are a couple of those blue peaks that are going up above that highest dotted line. So that means that these were considered major water flow events. And so since these are within that 6-week period, these might have contributed to the stranding. And so what we're going to do is, since we don't really know exactly how long it takes from when a seal is exposed to the Toxoplasma oocysts to when they develop illness with the disease Toxoplasmosis, we're going to look at different time frames within that red square, or within that 6 weeks prior to the stranding. And so then we're just going to go through and do that same sort of analysis with each one of our cases. And so, like, for example, here is RB24, a seal that stranded on the west side. And so you can see in that red box, the 6 weeks prior to RB24's stranding, you don't see any really major water flow events. You don't see any of the peaks going above that top dotted line. So in this case, there wouldn't be any evidence for a relationship to the increased water flow. But so we keep doing that. Our next case, we see some really large water flow peaks shortly before the stranding. And then here in 2018, we had a little cluster of a couple of cases, and indeed there were a lot of peaks in the water flow before those. And then in 2020, we also had a small cluster of a couple of cases, and there were some pretty big rainfall peaks before that. But of course, it's not just looking at the cases. This is why we do a case control study, because we want to look at other strandings of other types and see if maybe it just rains so much in Hawaii that this sort of thing always happens. So we'll go through and we'll look at the stream flow patterns before all of our control seals as well. And then we just need to do a little bit of statistical analysis to figure out if there is a meaningful relationship under all of this. And the analytical method that we're going to use is called an odds ratio analysis. So basically, this is essentially what you might want to do if you're a gambler. You want to know that you're getting the good odds of something happening. So you're essentially comparing the odds that a case is going to strand shortly after a major runoff event versus a control. And so in calculating the odds ratio, we make a, in computer land, we make something like this little square where we have major runoff events versus lower runoff events. And we're going to count that number of runoff events for our cases and for our controls. We'll count the number of lower runoff events for the cases and the controls. And then we can figure out if, in fact, the major runoff events are contributing to the toxoplasmosis cases. We would expect that number to be the biggest number. And so we're going to analyze this going back 1 week before the stranding, 2 weeks, 3 weeks, 4 weeks, 5 weeks, all the way up to 6 weeks. And we'll look at each week separately because, like I said, we don't know exactly what to expect with the timeline for the progression of illness. And so what we found was that, in fact, there was a much greater odds of heavy runoff events preceding toxoplasmosis cases. So here, this little graph just shows the odds ratios for each week. And so you can see 2 weeks prior to stranding, this odds ratio of 3.6 says the odds were 3.6 times greater that a toxoplasmosis case would have a heavy runoff event 2 weeks before stranding versus a control. And then when we look at 3 weeks before stranding, this peak we have here with a star above it, that was actually 12 times greater odds that a toxoplasmosis case would have a heavy runoff event 3 weeks prior to stranding versus a control. So this was a pretty significant finding and a statistically significant finding that really kind of showed, okay, these couple of weeks, these 2 to 3 weeks prior to stranding, it's much more likely that a toxoplasmosis case is associated with heavy runoff events versus any just other type of stranding. So this was pretty strong evidence for this. So some of our conclusions from this initial portion of our research is first and foremost that heavy runoff events do appear to increase the risk of toxoplasmosis cases. And this finding in Hawaii is really not a new earth-shaking finding. It's pretty similar to what others have found in other ecosystems. This is very similar to what they found on the West Coast, looking at risk factors for toxoplasmosis cases in sea otters. In New Zealand, they've also found that their dolphins are more exposed to toxoplasmosis in their rainy season. So, I mean, this is really something that we expect to find because we expect that as there's more rainfall, that can wash the Toxoplasma oocysts from wherever they're deposited on the landscape, and then that runoff can carry it into the marine environment where seals become exposed. So an expected finding, but still a very important one and an important thing to demonstrate for our ecosystem here, because for one thing, that supports the role of surface runoff in the oocyst transport and exposure. And so that can help us figure out or start thinking about some next steps in terms of thinking about what areas might have any higher risk than others. But also a really important thing that this does is it demonstrates a pretty local link to the pathogen source, right? So this tells us that local rainfall, local runoff are washing oocysts from our island landscape into our local waters. So this isn't, something that is mysteriously circulating around the entire Pacific Basin and reaching monk seals who knows how. This actually tells us that we do know how it's reaching monk seals, and that means we have some idea where it's coming from. It's coming from our own landscape, which again is not surprising, but it tells us that this is a problem in our own backyard, and that therefore this is a problem that we can take some management action to do something about. So it can be a little bit daunting, but in a way, this gives us some hope that because this is a local problem, we can try to find some local solutions. And then just one of the other really interesting findings was seeing that increase in the probability of the runoff events sort of peaking at the 3-week mark. It's not very many cases, it's not very conclusive, but it at least makes us start thinking about, well, maybe something about that 3-week mark is important or at least maybe gives us some clues about the normal progression of disease. So learning quite a lot from a relatively simple analysis to start out with. But then for what's next, of course, we're going to go beyond the simple and we're going to make it much more complicated. So our first analysis, just looking at the rainfall, really just sort of started connecting one of the legs in this epidemiological triangle. But next, we really want to incorporate all of these factors, the pathogen, the environment, the host, and we're going to use some more complicated models to try to understand the risk of toxoplasmosis across the landscape in the Hawaiian Islands. And so in doing that, there's a number of pieces that a model like this is going to involve. And so these are things we're just starting to put the pieces together. So I'm just going to give you guys a little overview today. I don't have the final results of this one to share with you yet. But so first we'll start with the pathogen side of the story. And so we'll have to calculate what's called Oasis Loading, or basically how much oocyst contamination is out on the landscape. And in order to do this, we'll have to sort of estimate how many cats or what densities of cats are on the landscape. And in order to do that, we'll think of cats as a few different types. So pet cats, and they might be associated with, human households. So in our land cover map over here, that would be these gray areas that are developed. And then we might have the colony cats, so the ones that get fed. And they're probably also going to be mostly associated with these gray developed areas, right? Because the cat colonies tend to be maybe at parking lots or trailheads where people can get easy access to feed them. And then you have neighborhood stray cats that maybe are not people's pets. They're not necessarily being fed as a colony, but these are the cats that might be sneaking a meal from a dumpster, other things where they're sort of associated with people, even though they're not pet cats that are owned and taken care of. And they might be primarily in the gray area. They might be a little bit out into the some of the agricultural areas in the yellow here on our land cover map. And then the last category is the wildlands, feral cats. And so these are the cats that are not really associated with people at all. They're living out in the wild, getting their own prey in nature, and they're going to be living mostly in these wildlands. So like the dark green and the light green, the sort of grasslands and forest lands on Oahu. So you can start to see how by thinking about the different types of cats, we can think about how they might be distributed on the map, because the different cats are going to be associated differently with different land cover types. And then they're also going to be associated with household density differently. So over here on the other side is just the census data from the US Census. And so we can use that to try to calculate how many cats given that we have some information about how many cats per household, then we can look at where the households are, and so where cats might be more and less dense on the landscape, which can give us an idea of how much oocysts might be contaminating different parts of the landscape. And then next, we'll look at the environmental factors, trying to figure out how these oocysts can be transported from wherever they're deposited on the landscape into the marine environment. And so we already have our first study that's, handy to say, yes, we're pretty sure that runoff is important. But so in the next step, we're going to use a fancier hydrological model to really look at what factors are impacting how much runoff is coming from different areas. And so this model is going to take into account the elevation across the landscape, stream flow, much like the data that we just used in this first study, the amount of rainfall in different areas, as well as the different land cover and how impervious different landscapes might be to the water flow, and how much of that water might stay on the landscape versus how quickly it might run off and carry oocysts with it. And so this model will basically treat the oocysts, since they're so small and microscopic, it will treat them like dissolved nutrients. So kind of like there might be hydrological models that look at how nitrogen is carried from agricultural fields into the water to impact coral reefs. This is going to look at how oocysts are carried from different parts of the landscape into the water to affect monk seals. So we've kind of talked some about the emergency response and what we can and cannot accomplish with emergency response. We've talked about some of the research that we've done so far and a lot of research that is coming up in the near future. So then the next tool to really talk about is the management and some of the management actions that we are taking in regards to mitigating the threat of toxoplasmosis. In talking about risk mitigation and management for toxoplasmosis, I'm really just going to be talking about a few of the things that NOAA is doing. Because keep in mind, as an agency, our purview is in the ocean, right? Whereas a lot of the source for this disease is actually coming from the terrestrial environment. So the management picture is much bigger than just NOAA, but obviously having an interest and a responsibility for the recovery of Hawaiian monk seals, we certainly have a role to play in the bigger picture of the management. And so, so the first thing is to use some of those scientific contributions to make sure that we are making the most scientifically informed, solid management decisions that we can. And we use some of this science and specifically apply it so that we can think about the management options that we have. So the whole purpose of this, the hydrological model that I was just telling you about, so that we can look at how oocysts are being deposited on the landscape, transported into the marine environment, and then encountered by monk seals, is so that once we kind of have something worked out where we kind of understand how that system works, then we can use that to sort of simulate different management scenarios to see how they might turn out, so that we can weigh different options using the model. Even before we try things out, we can use this sort of model simulation to think about how it might go. And then, of course, one of the other important roles that science plays is just maintaining some of the health and monitoring activities that we do, so that we have up-to-date data on things like how many seals are getting toxoplasmosis, what those cases might be related to, and things like that. And then, of course, some folks in parts of NOAA are involved in social science research that can be really informative in thinking about some of the views and relationships that people have with cats, and what we might think about in terms of managing those cats best. And so that's kind of part of where we get the idea of splitting cats into the different groups. Like, not only does that make sense on the landscape, but it also makes sense with the way people think about cats and the way you might be able to manage different cat populations. And so then the other piece of management really comes down to communication. So like I said, NOAA is not going to manage the entire landscape level problem of toxoplasmosis on our own as an agency. But so we're involved in a number of interagency working groups that are making some of those connections and having some of the conversations, so that we can come together as multiple agencies to really tackle some of these issues more holistically, and different folks in different agencies, different parts of the scientific community can kind of do their part for managing this situation. And then as far as within our own agency, we're also looking at sort of strategic communications around sharing our science, sharing the information, and talking about some of the management options that are out there. And then in the bigger picture, we're actually in the process of developing a strategic plan for looking at management options for toxoplasmosis, and really breaking down some of the things that our agency can do versus what we might need other partners and other agencies to do. And then, of course, the final question is, what can you do to mitigate risks of toxoplasmosis for monk seals and other animals in the environment? And the excellent answer is you can actually do something really helpful. So since cats play such a key role in this disease, simply being a really responsible cat owner, keeping cats indoors, keeping them safe, keeping them healthy, keeping them protected indoors is one of the most important things that people can do to protect the Hawaiian environment and Hawaiian species from this disease. If you have a cat, definitely dispose of cat feces and litter in the trash. We don't want to use the flushable litters, especially in an island environment here where some of that wastewater can make its way into the ocean. And then, of course, if you do see monk seals that are sick or injured or exhibiting that logging behavior, definitely call the Marine Mammal Stranding Hotline at 888-256-9840, and you can help us out by helping us get to those animals in need as quickly as possible. And as long as there is plenty of time, I will hang out and answer all of the questions that I can. And I just want to give a really big mahalo to you guys for showing up and sharing this Thursday evening to learn something about monk seals. And I want to give a big thank you to all of the other folks that have helped along the way with this research as well. So thank you guys. And then thanks everybody for listening. Hopefully it's less painful for you to listen to me than it is for me to listen to me. Yeah, let me know if anyone has any questions about monk seals and toxoplasmosis or other things about monk seals. I can try to answer them.
Hi, Stacie. I have a question. So when I looked at the information you just presented about there being a lag time between heavy rainfalls and presentation for with there being strandings associated with heavy rainfalls, with there being approximately a 3-week peak. That correlates really closely with the incubation period that's been reported in the literature with humans. And it seems to be, at least on the human side, there is a shorter incubation period with exposure to meat that is infected with toxo versus let me rephrase that, versus ingestion of oocysts. So with ingestion of oocysts, the incubation period is shorter. And with ingestion of meat, the incubation period is about 3 weeks, roughly. 10 to 23 days, with the median being somewhere in the neighborhood of 19 days. At least that was the one summary I looked at that reviewed, I want to say it was like 6 papers. Have you guys looked at any of the correlation between incubation period in other species and the potential impacts or incubation period in marine mammals?
So there's not a ton known about the incubation periods, and those are really great questions. I think the issue is often with marine animals, we don't understand that they have come in contact with toxoplasmosis until they become sick and strand. So we don't usually have much to go on to kind of work backwards to when they might have been exposed. So that's something, that we're trying to sort of get some clues about with this analysis, working back kind of week by week. I think we're starting to, like, that 3-week peak is really interesting, but again, with as few samples as we have, like, definitely nothing definitive about that. But one of the things that I would say, as far as thinking about whether seals might be getting exposed to oocysts versus the cysts that can occur in tissues, the cysts in tissues are generally occurring in warm-blooded animals, which for the most part are not showing up as part of the monk seal diet. Like since monk seals are eating fish, maybe some shellfish, crustaceans, we wouldn't necessarily expect that their prey items would be animals that themselves become infected with the toxoplasmosis, but they could very well be, filtering either fish breathing through their gills, or if they're eating some animals that are filter feeders. A lot of their prey items are probably accumulating oocysts and kind of carrying them along to the monk seals, but they're probably not infected themselves. So we are presuming that seals are getting infected primarily through the oocyst route, but whether they're getting that just directly from the water or eating the oocysts along with their prey is pretty unknown. Yeah, those are great questions and things that we're hoping we can learn some more about.
Thank you very much.
Mm-hmm.
Anybody else got any questions? I got a question. So I know most of the, I guess, toxoplasmosis is from cats. Have you guys done any studies on, because the runoff, it comes from the streams and whatnot. There's a lot of wild animals. Have you guys done, I guess, tests on other animals that could possibly be carriers of that versus cats?
Yeah, there's a little bit out there. Not a ton on Oahu. So I guess one important distinction to make is other animals can become infected with toxoplasmosis. And so like the cysts Mika was just talking about, they get cysts in their body tissue. So like, for example, humans, often the main route of getting toxoplasmosis is from undercooked meat. So like especially pork can have toxoplasmosis in it. And so that's the way that those other sort of non-cat organisms can pass toxoplasmosis. So only cats can defecate the oocysts in their feces. So only cats can sort of sprinkle the oocysts all over the environment. So the other animals are just holding those cysts within their tissues themselves. So it's not going to move through the environment in the same way. But those other animals can give us a really good indication of sort of how much toxoplasmosis might be out on the landscape. And so a really cool study that just recently came out, if anyone's interested, email me later. I'm going to forget the journal and everything at the moment. But just came out with some folks on Kauai using chickens as sentinels. So, it can be kind of unpopular to start trapping cats and people wonder what you're doing with cats. But chickens are also sort of using a lot of the same habitats. They're scratching and eating things out of the ground. So if there are oocysts in the environment, chickens are pretty likely to pick them up and eat them. And then, of course, they might have those toxoplasmosis within their tissues. And it's a little more feasible to convince people that you should go and trap their urban chickens, because sometimes people want those to go away and not be squawking all night and everything, right? So they trapped chickens throughout a number of different areas around Kauai and looked, I forget the exact numbers, but kind of a majority of the areas where they trapped chickens, they did find signs of toxoplasma. And there was a little bit of a signal with I think closer to the coast, they were finding even more toxoplasma than in the other areas, which, makes sense with more people closer to the coast, more cats around where the people are. So probably more oocysts in those areas. So yeah, so the other animals can be a really good indicator of what's happening on the landscape. So if you can't necessarily get all the cats, you might be able to do some mouse trapping, chicken trapping, looking at other stuff.
Cool.
Yeah, thanks.
Anybody else got any questions? I'm just gonna leave it open. Maybe for a few minutes. If we don't have any response, then I wrap it up. You want to mention your contact info?
Sure. So you guys take a look at the chat window. So I did put my email up there in the chat, but basically it is stacie.robinson@noaa.gov. So definitely, if you want to talk any more about toxoplasmosis or have other questions about monk seals, feel free to email me. We also have some links up there for the Hawaiian Monk Seal Program research Facebook page and also our NOAA website. And you can click through basic information about the species and also look at all of our news postings and web stories and cool stuff like that on the NOAA page.
Cool. I don't hear anybody asking any questions. So I'll probably wrap it up. Stacie for joining us. Hope you guys all can make it for next week, Thursday, October 29th. We actually have two speakers that we're doing one presentation. It's called Meet Your Neighbor Island Hawaiian Monk Seal Recovery Programs. It'll be Nicole Davis and Jamie Thompson for the NOAA Marine Mammal Response Network Island coordinators. So I hope you guys make it for that one. And thank you again, Stacie, and you guys have a great night and aloha.
Thank you, Gavin. Thank you, everyone, for joining us.